Okay. Hello guys. My name is Mahmoud Sadeghi. I am from Houston, Texas. I go by he, him pronouns, and I am a co-founder of BIPOC Neuro, which is an organization dedicated to representing minorities within the field of neuroscience and allowing more access educationally for those who are interested in the field or who don't know much about the field. Yeah, that's pretty much my mini bio. I guess I'll pass it on to Adrita.
Hey, hello, my name is Ajita Rahman, and I am a rising junior in high school. My pronouns are she, they, and I am another co-founder, just like Mahmoud. We're both from Houston, Texas, and it's nice to meet everyone, and I will pass it on to Alexa.
Hi, my name is Alexa. I'm also from Houston, Texas. I'm part of the BIPOC Neuro group, and I am going into my junior year just like Adrita. And Steph, you can take it from here.
Sure. I'm Stephanie Flavin. I work in HR at Biogen, and we've been working with the BIPOC Neuro team for the past few months, and we're really excited to be able to kick off the speaker series and help with the BIPOC Neuro mission. So with that, I'll pass it to Mercedes, and I'll do the full bio before you speak, but just a quick intro.
Sure. My name is Mercedes Baina, and I've also been interacting along with Steph with BIPOC Neuro for a few months now, trying to get this off the ground. This is our inaugural webinar, and we're so happy to be here today.
And my name is Sarah Stankowicz. I'm a scientist at Biogen, and I'm really excited to be involved with such an awesome organization, and excited to talk to you all about what neuroscience is for this first seminar.
Okay. With that, should we jump right into the presentations? Okay. So Mercedes, while you're getting your slides up, I'll do a quick bio here. Mercedes Baina. She's a researcher at Biogen and focuses on drug development for neurodegenerative disorders such as Alzheimer's disease. Captivated by neuroscience, she's worked in the field for over a decade in both academic and industrial laboratory settings. Mercedes earned her undergraduate degree in biology at Binghamton University and master's degree in biology from New York University. She's currently training for her 4th marathon, which will be in Berlin in September. I'll turn it over to you.
Thank you, Steph. Actually, I've been in neuroscience for 20 years now. And I've always known that I wanted to be in neuroscience because growing up, it was clear to me that members of my family suffer mental illness, it's brain disorders. And so I've always known that I wanted to do something with the brain and somehow try to fix it. So this is my way of contributing. And again, I want to reiterate, welcome students, because I realize many of you are on your summer break and you might be off doing some other summer activity like swimming. Instead, you choose to be here in this webinar talking about a topic that's near and dear to my heart. And so that tells me a lot about you, that you are interested in neuroscience, eager about neuroscience, or at the very least curious. So I'd like to say, fledgling neuroscientists, welcome. And with that, I'll overview what I want to cover in the next 20 minutes. I'd like to cover basic concepts in neuroscience. And then I'd like to speak about, well, basic concepts would be like, neuroscience is the study of the nervous system. And what exactly is the nervous system? It's the part of you that controls everything you do. The fact that you walk, you talk, what you think, how you think, what you remember. It also controls the parts that you don't do voluntarily. It's controlling the fact that your body's breathing for you all the time, and that you might be shivering in the cold or sweaty in the heat. And then I'm going to cover why I think neuroscience is so exciting. It's such a vast field. I kid you not, if you were to ask 20 neuroscientists the same question, what is neuroscience? You're going to get 20 unique answers. And then I want to end by just showing you some resources on how you can dive further into neuroscience right now, and then ideas on how to start a career in neuroscience. And so with that, let's dive in. The nervous system is made up of two big portions. The central nervous system, which is essentially your brain and spinal cord. That's the CNS, central nervous system. And then it's also made up of the peripheral nervous system, which is all the nerves that connect to the central nervous system. And the nervous system is made up of many cell types, but the rock star of the nervous system is the neuron. And even within neurons, there's many, many types of neurons. But what they all have in common is that they're masters of communication. And what makes them masters of communication is tied up in their shape. So here's the anatomy of a neuron. It consists of a cell body, what we call the soma, which houses the nucleus. That's where your genes live. That's where the DNA is. The cell body is connected to these tentacle-like things called dendrites. And what dendrites do is they sample the environment, they pick up information. So for example, a neuron that might be in your nose, an olfactory neuron, and don't worry, that's just a fancy word for smell. An olfactory neuron is going to pick up any little particles that are airborne that dissolve into the mucus of your nose, and then that is going to be somehow relayed to your brain to say, what exactly am I smelling? Is it a rose or is it dog poop? And then that information is sent to your brain in a magical way. It's literally sent with electricity. It's sent down this, I don't know if you see my mouse moving. Can you see? Okay, thank you. So that electrical signal or electrical impulse, the direction of the information goes from the dendrites to this axon. And I think of the axon as just any old electricity cord. So here I have my cord that I use to charge my phone. And inside the cord, the electricity is in wires and it's moving through. And then outside, the cord is wrapped with plastic, here white. And the plastic is insulating the cord so that the electricity stays inside the wires and not just dissipate willy-nilly and shock you. And the neurons has the same kind of concept. The neuronal axon is wrapped with this fatty substance. Here I'm showing, it's called a myelin sheath. So that makes sure that all the electrical signal stays inside that axon and goes straight away to the end of that axon, the terminals. And then the electricity has to do something, but we'll stop there for now because I want to share something quite remarkable about the neuron. The size of the cell body dictates how long this axon can be. The size of the cell body has a large charge. Just think about it as your cell phone battery. It's charged and it can last a full day because of how much battery charge can be there. Well, the cell body is charged and it can send a signal that is 10,000 times the width of this cell body. So to put this into context, I know that BIPOC Neuro is in Pasadena, your headquarters. So hi guys, I went and found a map of where you guys are situated. And there's a ring road, Beltway 8, which I circled. And then I said to myself, what if I land this huge neuron right inside this circle? The circle is 40 kilometers wide. And yes, I'm using metric system because in science, we speak in metric. So I'm not saying miles, it's 40 kilometers. How far do you think that this neuron can extend its axon? Can you type it in the chat? Is it going to extend to Boston where I am? Or is this axon going to extend to China maybe? How far do you think it's going to go? I can't see the chat. Is there anything? Mahmoud?
Someone said Boston.
Okay.
The entire Earth.
10 times wrapping the Earth. Can you believe that? And that neuron, so thank you for saying wrapping the Earth, you're on the right track, but tenfold, that's amazing to me. And the speed is 150 meters per second. I did the math in miles per hour. That's something faster than 300 miles an hour. I've never driven that fast. I've never been in a car that fast. Have you? Well, don't get any ideas. You'll be breaking the speed limit. So moving right along, when the electricity gets to the axon terminal, neurons don't actually touch, but they need to pass that information over to the next neuron in line. And the first time I saw a neuron, I thought that it looked like an octopus. So that's what I put here. Two little octopuses talking to each other, but they're not physically linked. They're close, but they're not touching. So there's this gap, and this electrical signal can't jump the gap. So the electricity is converted into molecular signal, a chemical signal. Many of you might have heard of neurotransmitter or dopamine. It's a neurotransmitter. Don't worry, it's just a fancy name for this chemical that so electricity comes up into these bubbles, and these chemicals are what jumps this gap to the next neuron. And so this gap where that information has to jump is called a synapse. And once many neurons are linked up in line doing the same job, they are firing together, so they wire together, and they think of them as getting the same color. That's called a neural circuit. So many neurons doing the same job, trying to do the same information. That's a neural circuit. So far we have covered, the nervous system is made up of, I was hoping people could put this in the chat really quickly, made up of, I don't see the chat.
Neurons.
Yeah, made up of neurons, thank you. And neurons specialize in? Put in the chat. Communication. Maybe people aren't fast at typing, or maybe I'm not being clear. I don't know which one, but I'm going to move on. And they specialize in communication using electricity and, oh wait, I gave an example, neurotransmitters. Thank you. Why, Pooja, thank you so much. And the last one is many neurons speaking together on the same topic form a put in the chat for me. Very close. It is a neural connection and a neural circuit. Thanks, guys. Thanks for playing along. So I will move along and change gears a little bit and talk about neurodevelopment. So all of these neurons, they're being developed when the fetus is being developed inside the womb. All of these neurons are starting to be generated, to grow, and they're present. So when a baby is born, let me ask you, how many neurons were you born with? Is it 10? Is it 20? 100? Put some guesses. 50, a billion. Oh, here we go. Thank you. Billion is on the right track. It's more on the order of 100 billion. I find that really crazy. A baby is born with 100 billion neurons, and those are essentially the same neurons that you will keep for your entire life in general. However, the baby doesn't have the ability to do what a child does. They don't walk, they don't talk. And an adult has even more ability than what a teenager might have. And so even though we all have the same number of neurons, what we don't have is the same level of connectivity. So it's as we learn, as we grow, as we get more experience, that's what our brain function gets better with learning and connectivity. And every time we get more connectivity, we get stronger and stronger circuits. So I think I wanted to ask, so one of the things that people do I think at teenager, adult age is learning to drive. And if Mahmoud, you put it in the chat, maybe I'll see if that could be a great theme that I could continue with. So if you put the poll.
Yes, I will launch the poll right now.
Thank you, Mahmoud. So a great learning step as we're getting to an adult is learning to drive. And so it looks like I'm getting people in all categories. So yes, I will go through with this theme of learning to drive. I think we can end the poll. We got more than 50%. Thank you, Mahmoud. I am closing the poll on my screen and I am moving forward. So one thing about development though, is that I think that at our society at age 18 might consider we're adults. At 21, society might consider us adults. That's one choice. But what age do you think neuroscience evidence is that we are adults? And I'll give you the answer. It's age 25. We don't get full maturity in an adult brain until we're 25 years old. And the brain develops from the back to front. So here I'm showing you the brain from the side. And these are the structures that we'll see if we just look straight, open your brain, your skull and just look straight at the brain. And so the last thing, so the back is the occipital lobe, and the most front part is the frontal lobe. And the last part to mature is called the prefrontal cortex. And this is where we have what's called executive functions. It's the roles for planning and impulse control. So whenever you are asked to do an assignment, maybe I'll say, okay, write an essay at the end of this talk. What have you learned? And then you will sit down. Don't worry, this is optional. I'm not asking you to do this, but you'll sit down and you'll start planning what the beginning, the middle, the end. And that's the type of executive function. And here's a view of, so I was showing before the lateral view. You can see, you can look at the brain from the top, the superior view or sagittal cut. Don't worry, you don't have to remember these names. It's essentially looking at the brain from the top, the side, or cutting it down the midline and then looking at it in the inside. That's the sagittal cut. And these in the colored regions are called the neocortex. The neocortex is unique to mammals. And what are mammals? You can put it in the chat, or let me ask you this, is a bird a mammal? Yes or no? Mammals are like dogs, dolphins. Just tell me yes or no, is a bird a mammal? Cats are mammals, humans are thank you. They're not a mammal, thank you. Because mammals are creatures that naturally will feed their young with milk. And that's how birds don't qualify. But all mammals have this colorful part in common. But what other creatures that are not mammals will have are the subcortical structures. And they're still super important in humans, but they will be more in common with maybe reptiles, right? So here again, I'm showing some big names, the limbic system, the thalamus, the cerebellum. But in the story, the next few slides, I want to talk about driving. So I wanted you to know that these are going to come up. But essentially, these are areas of your brain that we're using for feeling and memory, the thalamus for your senses, the smelling that I talked about before, touch, and the cerebellum has big roles in movements. That's all I wanted to show. So here I am. I'm going to tell you about learning to drive and areas of the brain that light up or fire at the time. So if you were to start using a steering wheel, you would know your fingertips will feel right away whether or not it's a plastic steering wheel or a leather steering wheel or fabric steering wheel because of the sensory information that's coming in from your fingertips. So those dendrites in the fingertips are sensing what they're feeling, and they route it to your brain in the cortex somewhere. But everything all your senses essentially get routed through the thalamus. So that's why I'm highlighting the thalamus here. An intriguing thing is if you were to have some injury to your somatosensory cortex or some part of your cortex, you probably still can function in the world. But if you're injured your thalamus, that is instant coma, like complete loss of consciousness. So it's super, super important in us. And another intriguing thing too is if you were to damage an axon in your fingertip, that's the axon that's in the periphery. Fingertip will go numb, it's not working so well. Maybe a year from now, however, you'll get feeling again. Those axons will regenerate, they'll grow back, you'll be okay. If unfortunately that injury would happen in an axon in your spinal cord, those axons don't grow back. Spinal cord injury will be with you for life. You'll, wheelchair. So inquiring neuroscientists want to know, well, what's different between the PNS axon and the CNS axon? If we could figure that out, we could solve and get cures for spinal cord injury. So there's a lot of unknowns in neuroscience that we still need new bright minds to come into the field and start solving a lot of unknowns that are still around. So I remember distinctly when I had my first driving lessons. I got into the car. My driving instructor, Mr. Green, had his coffee. I smelled his coffee that was routed through my thalamus. He had his music blaring, and I want to ask in the chat, do you think I wanted that music on or off? It's the first time I'm learning to drive. This is sensory input. Sometimes I don't want all the sensory input. I'm seeing all the road signs. I have to remember all the things that yes, off the focus, definitely off. Thank you guys, I love all the responses. Yes, I am just so concentrated using this prefrontal cortex, the thing that I used to study, the thing that I used to help me write those essays. I'm now using it to remember all the instructions I need to drive, right? So that's what my prefrontal cortex is doing while my thalamus is taking in all the sensory information. And then I'm using my cerebellum to make sure that I do the movements for the steering wheel. And of course, I was in this red Mini Cooper. It was stick shift. I had to deal with clutch and changing the gears myself. So that was like movements all over. And for my legs and everything, that was my cerebellum helping the movement. And I remember the first time I got on these huge highways, maybe 4 lanes in one direction, 4 in the other direction, big tractor trailer next to me. I felt they might be coming into my car. I had all these feelings. The amygdala is part of the limbic system that routes fear. It's this almond-shaped area in the brain, in the limbic system. That's the fear that I might have. Or the hippocampus that was being activated as I'm doing all of this stuff. The hippocampus also is good for memory and is the seat of navigation. And so all of these things were being lit up in my brain, firing in my brain. And by the time I got home, I was fried. My prefrontal cortex just needed to rest. It was wiped out. Has that happened to you where you've probably been studying for an exam very hard and then you find pure exhaustion? Yes, you were working out your prefrontal cortex.
Mercedes,
Yes.
I will say we might need to wrap this up just for the Q&A, just in case. The next.
So we're 5 minutes in?
Yes.
Okay, so long story short, when you accomplish something, your circuits for reward are lit up. So this is I have 5 minutes?
No, the 5 minutes are up.
The 5 minutes are up.
Yes.
Okay, guys. So I will just like to say, I didn't talk about, we study neuroscience in many, many model systems. I study human neurons in a petri dish, but we learn a lot from all types of organisms. There's tons of areas of research. I've highlighted a few through my talk. My studies are in neurodegenerative disorders and Alzheimer's. And I did want to talk a little bit about this. Maybe we'll do it in the Q&A after. Good resources. And yes, please, I'll take your questions right now. Thank you.
right, if anyone has any questions, we could drop them in the chat and we'll read them out loud, or she can use these extra minutes to continue her presentation.
Thank you, Pooja. I see your comments. I appreciate that.
So one question we have is, does having different cortical structures offer different types of animal-specific advantages?
So we do have the human cortex is a little bit different in that we have such a bigger and stronger prefrontal cortex than all other mammals. And I think that brings us a lot of human unique functions. So even our closest relatives don't have language like we do. So I think definitely it's giving us different advantages. Some animals like dogs have so much better ability to smell. They live in a world that we don't even have a sense how to experience that.
Another question from Abby. She said, so I know you said spinal cord injuries are permanent, but what is the case when someone can get physical therapy?
So spinal cord injuries are permanent now. But right now we also have what's called machine brain interactions. And so sometimes I think right now there's research on how to kind of skip the injury. So from the brain to the leg using some kind of machine, or we call that neuroprosthesis. So those types of things, I don't know if I'm answering the question, that can help. Physical therapy at some level might help, but I don't know how much. If there's just no function in the neuron, then it's gonna be hard to fix.
So our last question, how do these neurons and glial cells help in understanding consciousness and cognitive neuroscience?
Yes, that's a very wide-ranging answer that I could give. And consciousness is I don't think the field even has an answer where consciousness starts and stops. I don't think the field even knows the answer to that. But for cognitive neuroscience, I can tell you very well that the glia are the support cells for neurons. And if the glia aren't working well, neurons will die off. And then the cognition that the neurons are very, very good at will also go away. So it's very important that all the cell types work together and stay healthy.
All right. Before we move on to Sarah, there was one last question. For people getting into neuroscience, where do you recommend to start learning the basics?
So to start learning the basics, I think you're already doing it right now. Find neuroscientists that you can speak with, real life mentors, and ask questions. And then there's a lot of resources on the internet right now, very credible resources I'm showing here, Society for Neuroscience, the Dana Foundation, the BNA. They have a lot of stuff you can read, things that lay people, so people who are not professionals can read, as well as, it's your summer, you might just feel better watching videos, as well as their excellent podcasts, my favorite, with real world stories and presented in a very exciting way, are Invisibilia and Hidden Brain.
All right, thank you, Mercedes, for your presentation. It was wonderful. We do have extra questions in the chat that we'll move on to the next Q&A. So I guess I'll pass it on to Sarah.
So the last thing I wanted to say is the world of neuroscience is at your fingertips, pun intended.
All right. So Sarah, while you're loading your slides, I'll give a quick intro here so everyone is familiar with your background. So Dr. Sarah Stankowicz is a neuroscientist at Biogen, where she uses mouse and rat models of neurological disease to understand how neural activity changes in the disease and how it might be altered to develop new therapies for patients. In her PhD at UCLA, she discovered a novel role for the neurotransmitter acetylcholine in making fear memories. In her postdoc at Children's Hospital and Harvard Medical School, she studied how stressful events change neural activity and lead to anxiety. Outside the lab, Sarah writes fiction novels, sings in a local choir, and bikes around Boston. And with that, I'll turn it over to you, Sarah.
Thanks for that intro. And it's such a pleasure to go after Mercedes and giving such a great introduction to the different aspects of the brain and the function. And so I think this talk will be complementary. I'm going to be bouncing around between some different topics. My sort of metaphor for the brain as a structure that does inputs, processing, and outputs. I'll go into how I became a neuroscientist, just some kind of points along my journey. I'll talk a little bit about how our sensory systems can deceive us in the sense of various visual and auditory illusions. And then I'll talk a little bit about the work that I did in my PhD to give you a sense of what a neuroscientific experiment kind of looks like from beginning to end. And then I'll, of course, open it up to any of your wide-ranging questions. I particularly love the consciousness question because that's such a compelling one for many people who go into neuroscience and such a challenging one to study, but I'm happy to talk about that as we go on. So feel free to, if anything's unclear, drop a question in the chat, or I'm happy to answer at the end. So just to start off, this image is a picture of the cortex. So that's one of the regions that Mercedes talked about. And it's in rainbow colors because this is from the brainbow mouse that was genetically engineered to have every.
The slides aren't advancing for me, or sorry.
Oh, sorry. I haven't advanced yet, but thank you. This is the background picture on the first slide, which people can see.
Yes.
Okay, great. So the brainbow mouse is sort of a combinatorial expression of different proteins that have these different hues, kind of like mixing a bunch of different paint colors. And it allows you to visualize these really complicated neural circuits that I'll talk a bit about in the talk. Also, they're just pretty. And here's the legal disclaimer page. So I'm a neuroscientist. That's my day job. And what really brings me to work each day is trying to figure out how the brain works, as well as trying to solve neurological diseases, come up with treatments for patients. But how I got there. So I guess the first thing that every neuroscientist that I've ever met has in common is curiosity, and perhaps a little bit of oddness. So this is me as a child, sampling my environment, getting some sensory inputs in a creative way. I also was interested in mice, so that was useful because now I use mice as my model organisms in my research. So this is a picture of me and my sister for Halloween. But I guess when I first became interested in neuroscience, it was through reading the Scientific American Mind, which is a division of Scientific American that I think is still running. But just a lot of different experiments across neuroscience. The one that really struck my fancy was one on synesthesia, which is when your senses can become intermingled in the brain, such as, a low-level example would be numbers always look a certain color. So like the number 3 would always be yellow and the number 4 would always be blue. But a sort of higher level one would be that certain words or certain kinds of ideas always have certain sensory impressions. So I found that fascinating and wanted to get into neuroscience. So I went to Brown University and got my undergraduate degree. I got to do research in human research as well as with mice and rats, and found that I really liked working in the model. So one of the questions in the chat was, how do you choose your model system? And part of it is the types of questions that you like to ask. You can only ask certain kinds of questions in certain model systems. And the questions that I had about how neurons connecting to each other lead to particular behaviors or particular like internal states. What does it feel like to be a mouse doing a particular task? Those kinds of questions were really tough to answer in humans, but a little bit easier to answer in mice and rats. So I went on to do my PhD at UCLA, and I'll talk a little bit about that research later in the talk. Here is my lab. So you don't do science in a vacuum. It's not just a single mad scientist in a closet somewhere. Science is super collaborative. You get to make lots of friends and learn a lot from other people who are doing science as well. This is an example of me giving a talk during my grad school career, kind of outreach like this as well. Here's a picture of me in lab. I came to Boston to do my postdoc on stress and anxiety, kind of continuing that mouse research on neural circuits. And now I work at Biogen to try to take that information and apply it to human neurological diseases. And so, as Mercedes said, neuroscientists have wide-ranging topics that they focus on, wide-ranging kind of concerns. It can be anywhere from the super low level of, I just need to know how this ion channel works. What's the chemical makeup? What's the physical structure of it? So that when that ion channel gets disrupted, we can kind of work on ways to change that shape, to alter that configuration, to fix it. So that's the super low level, all the way spanning to consciousness. How do we measure different aspects of consciousness? How do we classify individuals who are in particular comas as at different levels of consciousness? What happens when we fall asleep? So those kinds of questions can only be answered in humans in certain ways. And so here's a picture of the brain. And here are some questions that I just think are fun, and some of them have answers, some of them do not. But that kind of questioning atmosphere and not feeling like there are always definite answers to problems is part of what being a neuroscientist is. Because the field is still so new, the tools are still developing. We can't always, get these concrete answers for our questions. So what happens to our minds when we fall asleep? I was fascinated with this as a child, having these crazy dreams, and I still, to this day, it's a little unclear exactly where some of those really complicated dreams come from in the brain. What is muscle memory? So this is pretty well understood. We can talk about this if you're interested in it later. Why are some memories so powerful and others slip away? This really drove a lot of my research. All of my research before coming to Biogen was related to memory. How do blind people experience the world differently? I love that idea of what it's like to be another person with such a different experience. Music affecting us so strongly, right? This has to do with how it's processed in the brain. And the big one, where is me? Where's the individual within these neural circuits? And of course, now my current work, how can we prevent and treat brain diseases? And so brains connect to each other at synapses, as Mercedes talked about, so I'm not going to belabor this point too much. This is more of a review now. So here is an example neuron. The inputs coming into the dendrites are going to be chemical from other synapses. That information is electrical along the axon, and then you connect to the next neuron in the chain. And so what this leads to, though, is that all we have, right, are neurons connected to each other, but different parts of the brain have such different functions. And a lot of that difference in function comes from how they're wired, this different connectivity. So these are different connectivity from the rainbow mouse. Again, here's this picture, whoopsies. Here's this picture of the cortex. This is a picture of the cerebellum. And here is my favorite brain region, the hippocampus. Just such fascinating differences in connectivity and totally different function. And so I think of the brain, this is kind of my metaphor, as a system for taking in inputs, processing them, and controlling those outputs. So you can kind of classify everything the brain is doing in one of those buckets. And so I want you to participate in the chat, and some of this came from Mercedes's talk as well. What are some examples of inputs that might come into the brain? So you had a nice primer getting you ready for this. I'll give you a hint.
Someone said music.
Music, yes, yes. So that'd be, that's exactly an input that can come in. Anything else?
Smell, reading.
Reading, yep. So sight, smell, you guys are all on the right track. Great job. So the senses are the big ones, right? So we got seeing, hearing, smelling, tasting, touching, which are the 5 that we all learn about as children, but there are lots of other senses as well. One is the sense of pain, which is distinct from touch. Another that I find, it was crazy that we didn't learn about this in school, but proprioception is the sense of where your limbs are in space. So if you close your eyes, you can tell where your hands are, where your legs are. And that's a totally separate sense from touch. So those inputs are all coming in and going to they're getting routed to the particular parts of the cortex and other brain regions that are processing them. what's one example of processing that the brain might do? I mean, music was a good one, because turning sound into something that means something to music. But I'll just give some other examples. So learning and memory is one. Emotional processing, right? Some of these sensory inputs are going to mean something to us. Planning, like in the prefrontal cortex, some logic, and many, many others. And finally, we have outputs, which are our muscles. I can't see the slides very well. Let me move our faces. So moving around, breathing is another output, as Mercedes said, heart rate regulation, emotional expression on the faces, and of course, many, many others. So I'll just give an example of this, of what this process looks like. So here is an example of an input. This is, pixels on a screen, but it's my dog. And so the processing involved is that looks like a dog, it whines like a dog, smells like a dog, right? I'm getting all these sensory inputs that are then being combined in the parietal cortex, actually. There's this memory that I remember this dog, it's mine. There's an emotion, that love, and then there's some motor output, which in this case is petting, as you must do with a dog. So we're constantly doing this input processing output as we go through our daily lives, and these kinds of things can be dysregulated in neurological diseases. Or there's just illusions that our brain can play tricks on us. So here is an example of a visual illusion. So if you pick up your finger and put it against the screen right along the line at the horizon, you will see that those two boxes are the same color. And I'm not playing any fancy tricks here. They really are the same color. But there's no way you can logic your brain into seeing them that way. Even if you know they're the same color, they're going to look different. Another example of this kind of illusion is this one on the right, where the two orange dots are the same size. That can't be true, right? They look so different, at least to me. And the same kind of thing is going on in this illusion, where the two lines are the same size, the same length. And what these are all examples of is this gives us a kind of window into how the visual system is processing the environment. It's not just a video camera that's just passively recording. The takeaway is that the visual system is relative. So for the first illusion, we live in a world where shadows exist, right? And so instead of us needing to know exactly the hue of a thing, we need to know where those shadows are in order to find these shapes of objects. So that's why that first visual illusion exists. And the other ones are that we need to know how things in the environment are different from each other. And so there are visual heuristics that we use to make those choices. All right, so that's a visual illusion. Next, we're going to look at we're going to listen to an auditory illusion. So this will especially work well if you have headphones on.
Oh, hello there. How are you? Yes. You are here for the virtual haircut?
Close your eyes.
Yes, okay.
It's
I
even
will
more
go get
powerful.
Luigi. He will come and cut your hair. I am Manuel. Just stay right there. Luigi. 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 It's Manuel here.
All right, I'll stop this here. The takeaway from that illusion is that even though it's just coming out of your computer, it feels like you're in a space with those people moving around you. And that's because we use the relative loudness between our two ears in order to tell where things are in space. So you can capitalize on that and make these really compelling illusions. And finally, language also can cause us to have some sensory processing issues, which are, I think, also counts as an illusion. So I would like a volunteer from the audience. So you can volunteer all you're doing is reading words aloud or saying the color of things. So if you could just post in the chat, that would be awesome. Then we can unmute you. It's the Stroop test, if you've heard of it.
I believe Abby, well, we have two volunteers. Abby was the first.
Awesome.
So I'll allow her to unmute.
All right, thanks so much for volunteering, Abby. So to participate in this, first I would like you to just read the words across the top line. Starting with the word blue. Is the unmuting not working?
So it looks like Abby's deferring to Waipuja.
Okay.
Mahmoud, are you able to unmute?
Yes.
Do you want me to just read the words?
Yep, just read the first line, ignore the colors.
Blue, orange, green, red, purple.
Ooh. Okay, and read the second line again as fast as you can, ignoring the colors.
Red, purple, blue, orange, green.
Awesome, so that was perfect. That was super fast and great. So now the next line down, instead of reading the words, go as fast as you can and say what color the word is written in.
Purple, orange, red, blue, green.
Good. You want to do one more line?
Okay.
Bottom line.
Red, blue, green, orange, purple.
Nice. Okay, so the last line was a lot easier than the second to last line. Thank you so much for your participation. You did a great job. So the reason why the second to last line was hard is because there's conflict between what's written there and what you're trying to do, which is name the color. The last line's easy because there's no conflict. So you've got two systems going on, one that you're really used to doing, which is reading. We have a lot of practice in that. And the other one, which is naming colors, which you're not used to doing. So it's a lot harder.
I will say we have 5 minutes left.
Great, okay, right on target. So I'm gonna quickly go through my research from my PhD. So I studied anxiety and fear. And the whole purpose of anxiety and fear in the brain is to keep us safe, right? If there might be a threat out there in the world, you want a system that's going to be very quick to evaluate that threat and act such that you don't get eaten by a lion or equivalent. But the problem with these processes in the brain is that because there's such evolutionary pressure to have them be very effective in order to survive, you can end up with the systems going too far in individuals and with anxiety disorders or PTSD, for example. And so I'm talking about that on this slide as well, that anxiety and fear can overwhelm our rational brain. So Mercedes talked about the limbic system, and I'm showing those arrows coming from the limbic system, that something might be wrong out there in the world, and there's this powerful signal that's being activated from those regions. And the prefrontal cortex, for example, that's saying, hey, maybe we should take a deep breath and figure this out, can be quite quiet. It's not powerful enough to overwhelm that really intense subcortical activation. And so, as I said, when fear goes wrong, it can lead to PTSD. And you will be too young to remember this, but back when I was young, the two towers fell, and this led to a generation of many people who were afraid to go on planes, had anxiety about open spaces, and some of these symptoms still persist today, even with therapy. So there's not a known drug that we can just give to treat and get rid of PTSD, for example. And so this leads to my research from my PhD, is that we have to understand how fear memories are formed in the brain in order to eventually be able to change their hold over us in these disorders. So here's a picture of the mouse hippocampus. The neurons are in blue in this picture. And as Mercedes mentioned, the hippocampus is in the part of the brain that helps us navigate our environment and form specific memories of our lives. So this is a pretty ideal target for fear memory. I was drawn in my research to a specific chemical signal, neurotransmitter. This is the structure of acetylcholine. And acetylcholine increases when we explore a new place and decreases when we're recalling old memories. So I thought, what if acetylcholine is kind of the record button on our memory? And if we increase it, we can make a stronger memory. So with a little help from my research subjects, so this is a rat, I worked with both mice and rats. I set out to see if acetylcholine could make stronger memories. So first I explored allowed mice to explore a new cage. And in some of the mice, I increased acetylcholine in the hippocampus using lasers. And the next day I gave them a short time in the cage and gave them a mild foot shock. So this is usually, normal mice don't have enough time to recognize where they are, and so they make a very weak fear memory from this. And what I found was that the following day, only the mice that had the increased acetylcholine were afraid. And so this suggested that the acetylcholine 2 days ago, when they were learning about that environment, actually served to make that fear memory later stronger. And so this had implications for treating PTSD, and I followed up some of those in my postdoc. But now I've gone on to expand to different neurological diseases at Biogen by studying rodents and their neural circuits. And so I just added this slide because I'm working in rodents. We're trying to translate to humans, but in humans, we can't do invasive things. So all we can do is non-invasive techniques like the ones here. So MRI on the left allows you to see the structure of the brain. DTI allows you to see those axons. So that's the second picture, these what look like wires traveling all over the brain. And on the third is PET. So that allows you to see the metabolism of different kinds of chemicals in the brain. And finally, fMRI allows you to see activity in certain parts of the brain, and that's what's lit up in that picture. So my research is trying to record from mice, understand their neural circuits, and then try to translate to how human neural circuits change in disorders. And so to become a neuroscientist, as Mercedes had in her talk as well, learning these basic scientific principles, maybe computer science, more and more computer science is becoming a huge part of what neuroscientists do day to day, partly because we just have so much data and so much information that we really need computer science to make models of the brain and to process that information. Being curious, so you guys are already curious, you're here. Reading and watching videos of the brain is a great place to start, seeing what tickles your fancy, what kinds of parts of the brain you're interested in. That will help you figure out what kinds of research you might want to do. This is a big one, learning to make mistakes, forgiving yourself, and keep going. Every scientist that I've ever met has failures, right? You have experiments that don't work, sometimes for reasons that you can never figure out. And so being able to forgive yourself is so important. And just because an experiment fails doesn't mean you're a bad scientist. Just means you need to try again. And finding a mentor is a great way to also learn about the field. So we've got a few minutes left for questions, and I'm happy to take them. So thank you guys for your attention.
We have a question from Abby. How does synesthesia work? Or does it just automatically correlate certain colors and textures?
That's a good question. So it's still an area of active research, but my understanding is that during development, as Mercedes said, we're born with the billions of neurons. They're all connected up in these crazy ways, and part of growing up is pruning some of those connections. So you don't just want to create new ones, you want to get rid of connections that don't make sense. And so part of what they hypothesize is going on with people with synesthesia is that some of those early connections that normally get pruned are still persisting. So you might have connections between your visual cortex and your auditory cortex that the average person doesn't have, and that somehow, or within the visual cortex between color and number shape. And so what it's like for those people, I don't have synesthesia, but I've spoken to people that do, is that that's just part of their sensory experience. In the same way that when you guys looked at the two boxes with the shadow in between and you couldn't mentally tell yourself that those were the same color, synesthetes, when they're looking at a number, they just see it as a certain color. If they have number color synesthesia, there are many different kinds.
Thank you. Our next question is, does high stress lead to deletion of memories?
That's a good question. So stress is interesting because it seems like it's all about the level of stress. And this is a generalization. Things are going to vary depending on the types of stress and the different situations. So one variance in the type of stress is one really intense short stress, like the 9/11 terrorist attacks, as compared to really low level stress that persists for a long time. So those are two different kinds of stress, and they can lead to different changes in the brain. So, but in general, low levels of stress, like just a little bit of stress, will facilitate forming new memories, and will facilitate processing, because it kind of tunes your brain and gets it ready to run, ready to do stuff. But there's a level of stress above which it's going to make it very difficult to form new memories, to recall memories, because so much of your attention is focused on the stress that it's not actually helping any of the other functions. So there are different levels of that. There's kind of a sweet spot for just feeling a little uncomfortable when you're about to give a talk, for example, might make your talk better. But above a certain level, it's going to be impossible to give the talk.
All right. Next, we have, what is something you wish you had known before diving into this field?
The computer science aspect. I wish I had taken more computer science classes in college. It would have really helped me test some of my hypotheses in rigorous ways. So instead of going to the mouse, for example, and running a bunch of mice in experiments, you can come up with a model for how that mouse works and run it through a computer simulation 10,000 times for free, instead of using mice, for example. So I really like that kind of interplay between being able to model how these systems work and then test whether those models are accurate by running them in real humans or real other model organisms.
Next, we have, is anxiety beneficial or not? Because if it isn't, it should have been eliminated through the process of evolution.
Yes, it depends on the circumstance, right? So in a situation where the threat is real, anxiety is essential. If you don't have anxiety and there's really threats out there, that's going to lead to you not passing on your genes. The mouse gets eaten by the cat in that case. So that's why evolutionarily fear and anxiety are so well conserved, and you see it across all of these different kinds of organisms. I mean, we can study analogs of fear in worms because just even though they don't have all of these cortical structures and things, because some of these avoiding threats is so essential. In terms of our modern life, there are certainly a lot of situations where we wish we could just take a pill or turn a switch and turn off anxiety because we know that the threat's not real. And so part of research is trying to come up with therapies where we can either titrate down the strength of those fear memories or titrate up the control function. So like maybe make the prefrontal cortex stronger, such that when you do your therapy and you say, no, like there's not really a threat out there, the rest of your brain has to listen. So people are working on it.
And then there was one last question on, you discussed music briefly. Can you explain the phenomenon of earworms?
Earworms, where a song gets stuck in your head. I don't know that much about earworms, I have to say, although I suffer from them greatly. Yeah, anything would be guessing. Sorry.
We have another question. How does neurobiology of language and memory help in unlocking the consciousness?
Ooh, good question. Language and memory and consciousness. So when I think about consciousness, there are kind of a few different angles to the question. So one is the like sleeping to waking state, like when is someone conscious? Another would be, are very young children as conscious as an adult is? So there's like a spectrum of consciousness and development as well. And then finally, there's like consciousness of things in the world, where that's kind of an analog of attention. So like, can you show a picture in such a short amount of time that the person doesn't consciously see it? They can't tell you what's in the picture, but parts of their brain are processing it and having emotional responses to it, for example. So those things are not conscious, but they are being processed by the brain. And so those kinds of ways of thinking about consciousness help us to try to unpack what it means to be conscious. So your question was about language and memory. So I guess memory, how that intersects with consciousness in my mind would be we have so many memories that we can't consciously recall, but are still informing us and helping us move through the world. Like, for example, I talked a little bit about muscle memory. So playing soccer, I've played hundreds of soccer games, but I can't remember the experience of each one of those, but I still have muscle memory or the ability to play soccer. And so there are some kinds of memory like that that are not conscious recollection, but are still kind of informing our actions. So we can study those as well. I'm not really sure about language in terms of consciousness. I guess you could say, you know, having multiple languages and the ability to switch between them. You can see how people who are bilingual or trilingual or more, how their brains are different than somebody who just speaks a single language, and try to understand how that processing contributes to their conscious experience. I hope that was answer enough. It's a pretty big question.
Thank you, Sarah, for speaking and for answering our questions today. We really appreciate it.
Anytime.
So next we will be posting our survey link in the chat. Steph will put up a slide for some directions.
Sorry, I lost my mute button. Yep, so I believe, can everyone see the slide I put up?
Yes.
So Mahmoud, I believe, is putting a link in the chat with the survey for the speakers today and the experience today. So if you are able to complete that, that would be great, and have a chance to win a lovely Biogen water bottle.
The survey takes no more than, I'd say, 2 or 3 minutes to fill out, but we will also email the survey to attendees and those who registered so that they can also fill it out later on. And we'll leave it open for 1 or 2 days and then do a raffle.
Okay. Thank you everyone for your time today. This was great.
Yes, everyone, thank you for joining. Our next webinar will be in 2 weeks and we'll have new speakers and we hope to see you soon.
Thank you everyone for listening. Bye-bye.
Bye.